Hello and welcome to another one of D's ADD videos. Today we're going to be discussing action potentials and all the vocabulary and concepts associated with action potentials. So what is an action potential? Well, the membrane has a resting potential of negative 65 millivolts. Okay, watch the video on the membrane if you haven't seen it already to explain how that occurs. Basically, the sodium ions and calcium ions and chlorine ions are higher in concentration outside the cell, and the potassium ions are higher in concentration inside the cell. That asymmetry of ions creates a concentration gradient and also creates a potential difference of negative 65 millivolts. Again, all in the membrane video. But what happens is, in response to stimulus, there's little local potentials. So a little bit of sodium comes in, for instance, into the neuron and creates this little tiny jump in positive potential. These are all called little graded potentials, and they can add up either spatially or they can add up temporally over time. All this will be covered in detail, but basically once they add up and reach this threshold, then the switch is flipped for the action potential to start at the axon hillock, and we depolarize, meaning we become more positive, all the way up to positive 40 millivolts. And that's called the equilibrium potential for sodium channel. And basically what happens is we have no longer any current of sodium at this potential. The channel or the protein is shut down for business, and the movement of the sodium ions, no longer occurs. Whereas at that same potential, there's a trigger of turning on these voltage-gated potassium channels, which allows potassium to go out of the cell, efflux, and that whole thing right there is called hyperpolarization. Okay, so the first half is the depolarization, and due to sodium influx, whereas the second half is hyperpolarization and due to potassium efflux. So that's what this is saying. All right. So the membrane potential becomes more positive due to influx, meaning outside to inside of the neuronal membrane of, in this case, sodium ions. And then hyperpolarization is the other half, the second half of the action potential, and that's when we go from inside to outside, and this is of potassium ions. Okay, just in case you forgot the concentration relative to each other, outside we have a abundance of sodium, chlorine. I forgot to put calcium on there as well, so let's put calcium on. Same color, I guess. And calcium is big on the outside and itty bitty on the inside. And also small proteins goes out here outside the cell. There are charged proteins that help in this asymmetry of charge, creating that membrane potential of negative 65 millivolts. Okay, so that's the difference between hyperpolarization and depolarization. Hyper is potassium, D is sodium. And let's go through some of the details. I went kind of fast through. Okay, so what is a local potential? Well, if you remember your nerve, I think I drew a picture of a nerve, has all these dendrites out here. And these dendrites start picking up signals from whatever they're synapsed with, either another nerve or it could be some receptor of some sort, like a skin receptor for touch or temperature, whatever it is. And they sum up the signal by adding a little bit of sodium in response to however strong the signal stimulus is. So let's say we have a stimulus coming in here, and a little bit of sodium comes in, and what happens is there's a ripple, a kind of like throwing a rock into a lake. You get ripples. Well, there's a ripple of positive potential coming in, all those sodium ions. And then if there's another signal that comes in right there, stimulus, that ripple reinforces this ripple, okay? And you get a bigger ripple coming down this way. And what I just showed you there is a bunch of local potentials adding up, and they're adding up spatially, okay? Remember I told you there's two ways. There's temporally and spatially, and this is the spatially adding up. And multiple channels, or proteins, whatever you want to call them, are influxing sodium, and they're close enough in proximity that they end up adding in potential. Right? And once they add up enough, these little graded potentials here, if you remember when we discussed this earlier, we have all these individual little humps are the graded potentials, and they can sum up over space like I just showed you. Or what could happen as well is you can have a temporal one. So let's say this spot just keeps sending a stimulus over and over and over time. What happens is the action potential goes up a little bit from the first one, or the graded potential, sorry, and as it's starting to come back down, well, the next stimulus has come in, so it goes up a little bit more. And then as it starts to come back down, the next stimulus comes up in, and so forth, and keeps repeating the pattern until that threshold is reached. Then the actual action potential begins. All right? And that was temporally, we just looked at the summing up of the action potential temporally. Ooh, red highlighter. And it adds up over time. That's the key there. That didn't work out too well. On green, it makes it look black, but with the orange, let's try yellow. So, stimulus adds up over time at the same spot. Either way, what we're doing is we're going to add up to reach thresholds, okay? And then that switches the signal at the axon hillock to begin the first action potential. So what is the axon hillock? Well, it's the region of the cell body that protrudes out the axon, and basically it has a high concentration of sodium channels right there, what I'm highlighting. So, and these sodium channels are voltage gated. If you remember what that means is, in response to the increase in potential, they open up and allow sodium to come in. So what's happening here is these stimuli are in response to a receptor of some sort, or another nerve stimulating it, and it depolarizes, and all those ripples add up, whether spatially or temporally, and once it gets enough positive around this axon hillock from all those sodium ions coming in, then it flips the switch and it starts that action potential depolarization that we saw, and a bunch of sodium channels get activated at the same time. And what that does is it starts a really big wave that, a really big wave that travels down the length of the axon a little bit until it reaches a channel. So all these sodium channels open, and all these influx of sodium ions occurs here. Okay, this is after we've reached threshold at the axon hillock. And all these sodium ions have started to drift down the axon. Well, because they are more positive than what they were before, or what the membrane was before, we now make the inside of the membrane a little bit down the axon more positive. And what happens is these voltage gated channels open and then allow sodium to come in, making the membrane more positive. And then a little bit further down the line, there are going to be some more sodium channels that react, and basically takes that action potential and just propagates it down the axon until we get to this presynaptic button. And that's a whole different video there and what happens. Okay, so again, a bunch of sodium comes in through the axon hillock, and that sodium travels down kind of like a ripple, and that all that sodium coming in makes the axon a little bit down the line more positive potential till it reaches that threshold, and basically it causes these channels that are down the line to open and allow more sodium to come in, and then repeats the process over and over. Okay, you might have to watch this video a couple times to really get what's going on. Make sure you do your reading and ask any questions as they come along. Don't just not know what something is. It's better to ask. The only bad question is the one that you don't ask. So I hope I've cleared things up about the action potential. Thank you and have a nice day.